I see that my presentation is showing up. Okay, so from this presentation, I'm going to cover the contributions of plant breeding for sustainable food and agriculture, genotyping and phenotyping technologies, the prospects of genome breeding, the boost brought by deep learning to breeding with large data sets and complex traits. I'll be citing many public sources, and there is a list of references at the last slide. First, the Food and Agriculture Organization of the United Nations, FAO, has defined 5 key principles for sustainable food and agriculture: increasing productivity, employment, and value addition in food systems, protecting and enhancing natural resources, improving livelihoods and fostering inclusive economic growth, enhancing resilience of people, communities, and ecosystems, and adapting governance to new challenges. Aligned with these 5 principles, there are 20 interconnected actions to guide decision makers in transforming food agriculture to achieve zero hunger and the sustainable goals, development goals. Of the 20 actions, many are already addressed by plant breeding. For example, access to productive resources, diversification of production, protection of natural resources by breeding crops that improve adaptation and resilience to environmental changes, and improve soil health, water usage, and nutrition. Plant breeding continues to drive the adaptation of new technologies, as we will see in the next slides, that's not only strengthen innovation systems, but also lead to institutional reforms. And also support the diversification of agricultural systems for the protection of biodiversity and the enablement of circular economies. Over the last 50 years, global population has more than doubled, and that has inevitably reduced the land available per person to live and grow food. However, the continuing improvement of technologies developed during the Green Revolution of the late 50s and 60s allowed major increases in crop production and yield, with minimal expansion of land use since the late 70s. However, the Green Revolution is still unequal. Yield gains that are achieved by European and North American countries are still to be reached in South America and Asia, and seem a far distant reality for African countries. Nevertheless, most of the heavy lifting of expensing technology development has already been done by better served countries, reducing the overall costs to the point that these technologies can be adopted and improved to local needs of underserved regions and countries. One well-known example of technology cost reduction is genome sequencing. When the first human genome was sequenced 20 years ago, the process cost $2.7 billion and took 13 years. Today, it costs around $750 and can be done in a few days. Soon, it will cost only $100 to sequence your genome in a few hours. This reduction in cost and time of genotyping human genomes can be extrapolated to plant genomes, and is reflected in the number of plant genome assemblies publicly released in the past few years. Genome assemblies of 798 land plant species were made available as of January 2021. The advent of long-read sequencing technology associates with an increase in number of contigs sequence that allow better contiguity and quality of these assemblies. Given model-deployed species and species that are agriculturally and economically important, like maize, rice, and soy, have an over-representation on the number of the assemblies. However, with the improvements offered by long-read sequencing, it's becoming more feasible to sequence and assemble large polyploid plant genomes and genomes from orphan crops. They're not yet traded internationally or started to be recently traded like the allotetraploid quinoa. The availability of reference genome sequences, the cost-effective genotyping platforms, and a range of genetic populations have provided new, faster sequencing-based trait mapping approaches that allow breeding programs to map target trait markers in a time and cost-effective manner. Once markers are identified, high-throughput sequencing technologies allow genotyping of hundreds to thousands of markers in one pass and the screening of millions of new recombinant seeds in a time-effective manner to select those that have the desired marker combinations for planting and phenotyping in a breeding program. Once the genes and regulatory elements responsible for a trait are identified, genome editing allowed direct improvement of elite breeding lines or commercial varieties, eliminating years of back crosses and integration cycles relative to traditional crossbreeding or mutation and transgenic breeding. It needs to be pointed out, though, that the global adoption of genome editing technologies in plant breeding greatly depends on how governments regulate its use. It is still unclear in most countries, particularly those that would benefit most from adoption. Nevertheless, as genome editing technologies are widely accessible and relatively inexpensive to implement, projects are in progress to improve food security crops. This table on the right shows the crops, traits, and stages of product development from genome editing projects carried at the Consultative Group for International Agricultural Research, the CGIAR centers. Some of these projects actually receive technical and funding support from companies like Corteva. But despite all the advancements in genotyping, phenotyping at scale remains a challenge. So the selected genotypes need to have efficacy measured in greenhouse, field plots, field tests, and multiple locations to first select the winning varieties. There are several technologies that are increasing the throughput of quantitative phenotyping at ground, above ground, and below ground level that we will address next. In controlled environments, there are automation systems that bring pot-grown plants into phenotyping chambers with sensors that can measure spectra of light that correlate with plant physiological traits and can be used to model crop growth and predict crop health and yield. At field plots, there are autonomous rigs that carry those same sensors over plants. However, the speed at which the field scanalizer system operates is slow, the space is limited, and the throughput does not match the scale needed in a cost-efficient manner. Remote sensing can cover much larger areas. There has been a lot of investment on startups in this space in recent years, and some of them now have algorithms that provide actionable information, such as identifying trees in an orange orchard infected with holland bean or citrus greening disease before humans perceive visual symptoms show up that can take anywhere from 6 months to 2 years. Removing the infected trees as early as possible considerably minimizes the spread of the disease and the incurred economic losses. Proximal sensing technologies are catching up to remote sensing ones. At ground level, there are autonomous rover systems that carry sensors and run in between rows, capturing traits within the canopy that cannot be detected from above, like corn ear height and ear drooping. There is research in progress to develop in-field root phenotyping systems that are non-invasive and non-destructive to replace the traditional shovalomics method. But The challenges are more difficult due to interference from soil properties and the inverse correlation between the depth and the resolution at which thin roots can be imaged or even detected. For example, tubers like cassava can be easily imaged by these systems, but soil roots overlapping with each other are difficult to isolate and measure. Technologies that measure below-ground soil properties have been around for many years. The recent growth of carbon markets drove a boost in investment to extend their capabilities to measure soil organic carbon, bulk density, and carbon stock. Technologies that measure hyperlocal weather received a boost in investment in parallel with remote sensing technologies. Environmental and crop data from remote and proximal sensing are combined to improve crop growth models, yield forecasts, irrigation, and fertilizer management. Carbon markets may also drive development of simpler and cost-effective alternatives for equivariance flux towers. They are the current gold standards for measurement of greenhouse gas emissions. Studies show that the computational analysis of all the information captured from these imaging and sensing technologies can detect phenotypic differences that not only match with high or medium accuracy to hand measure phenotypes, like those in the left graph, but also that are latent space phenotypes. They're not visually evident to humans, but contribute to phenotypic responses and higher genotype. Adding cropping systems and input application management practices to genotype bioenvironment interaction analysis is challenging, but it's required to breed varieties that are sustainable and can secure yields under current and future climate scenarios. For example, the broad bulk long-term experiment with winter wheat at Rothamsted Research in the UK shows that the yield of the first wheat in 5-year rotations with break crops is almost double than the yield of continuous wheat fields, independent if synthetic fertilizers or manure was applied. Part of the reason is lower root disease pressure. Deep learning analytics of G by E by M, large and complex data sets, may identify other contributing factors and better guide breeding steps. Another challenge will be to breed beyond monocultures. Intercrops are known to have multiple sustainability benefits like increased soil health, biodiversity, and crop resilience. But there is reluctancy to adopt due to the complexity and perceived uncertainty in yield and profitability. Studies show, though, that the net gains with intercropping are positive, and the revenue from grain yields, saved costs on fertilizers, and the avoided health costs surpass the losses from additional machinery and labor costs. Governments around the world are promoting intercrop adoption. Carbon markets may also pay farmers for intercropping adoption once there are models that predict their induced increase in carbon sequestration. There are already studies proposing breeding program designs that are optimized for intercropping with multiple species that use high throughput sequencing, genotyping, phenotyping, and the analytical technologies that we reviewed in the previous slides. This schematic figure from Xu et al. Summarizes well the modern breeding technologies that we reviewed in this talk. The scale of operations is large and require many resources. But as we have heard through the symposium, there are several collaborations between academia, public sector institutions like Embrapa and the CGIAR, nonprofits like the Gay Foundation, and private companies like Corteva. Together, we share the knowledge and resources to address development of crop varieties under the Sustainable Food and Agriculture Framework. I encourage you to visit Corteva's Open Innovation website, where you can see examples of collaborations we had, the type of resources we can provide, our areas of interest, and submit proposals for collaborations.